Hi guys, so welcome back. Today we're going to start chapter 11 or Mendelian genetics. So this is going to piggyback a little bit off of chapter 12 where we talked about the chromosomal basis of inheritance, aka meiosis. Mendelian genetics is likely something that you've encountered in other classes. You can kind of think of this in a way as Punnett square genetics. So what we're going to be doing in this chapter is of course reviewing Punnett squares, but also learning what we can get from these Mendelian ratios and how we're going to be able to lay a foundation using these ratios to help us do more advanced genetics in the upcoming chapters. So let's go ahead and get started with just kind of a review of what we're going to be looking at in this chapter. So of course, we're thinking about all the time as geneticists this relationship between physical traits or the phenotype and the underlying genotype. So we're always trying to figure out what contribution the genotype is going to have for giving us a particular appearance or trait. Of course, there are some environmental influences and some randomness involved, but what we're really interested in is how much we can connect the genotype with the phenotype. So that will be an underlying theme throughout the semester. So of course, when we think about Mendelian genetics, we have to start with Gregor Mendel, and he was able to come up with these ratios by using variation in pea plants to study their genetics. So we look at 7 different traits: the seed coat flower color, seed shape. We're looking at the pod color, pod shape, the stem height, and the flower position. So we won't be looking at all of these at once, but we'll be talking about some of the elements that we need to make this work and how these were actually studied. So these crosses were performed by Mendel, and they weren't just random. These weren't just like putting pea plants out in a garden. They were very controlled. So for example, we have, of course, flowers, phenotype 1 and phenotype 2. It's important to use contrasting phenotypes here. And plants, as we know, have largely both female and male organs, so they're able to, in a lot of situations, they can pollinate themselves, etc. But we don't want that to happen. We want to be able to control this cross in a really specific way. So we are then going to actually remove stamens to create what we call female plants. So this is called hand emasculation, and I've actually done a bit of this in my work with sorghum to actually remove the anther, so we only have female parts on our plant. Okay, and so then we can collect pollen from the male plant. So I think this is actually the female plant over here. We remove the stamens from the female plant, leaving only the pistil. We then take pollen from our male plant and specifically pollinate this female. So we'll actually sprinkle a little bit of pollen, and then we know that if this particular cross results in offspring, that it will be specifically a cross between these 2 plants. We then look at the development of the peas, and we observe the offspring. This is the phenotypes of the offspring that we're interested in. This is also called phenotyping when we're recording the phenotypes that we're interested in. So again, controlling these crosses really specifically. So of course, observing these offspring can give us information. So just a couple of things to look at so that we're all on the same page with terminology moving forward. So of course, there's the P generation that stands for parent. Of course, the parent is relative because everything that we know of has a parent, but these are the ones that we're kind of starting with. You can see contrasting phenotypes, smooth seeds versus wrinkled seeds. We cross them, and in a normal relationship, we understand how recessive and dominant traits work. In our F1 or filial 1, this is the first generation, all the seeds are smooth. So there's not a picture here, but they're all smooth. This would tell you a couple of things. Then when we take these F1s, there are multiple plants in this generation, basically crossing brothers and sisters, F1 by F1 cross. This is where we're able to see these Mendelian ratios come out. So Mendel deduced that these phenotypic differences we see, these smooth and wrinkled seeds, this trait, are due to alleles. So now we're kind of thinking about the actual underlying thing going on. And these alleles remain distinct. One of the traits, being the recessive trait, can be masked in the F1 generation. And then when we cross F1 by F1, of course, that's when we're able to, again, reveal that recessive trait. And this is Mendel's principle of dominance. So this is where you see that 3-to-1 ratio. I'd like to also point out, look how many plants were tested here and how many seeds we're actually looking at. Doing genetic analysis often takes thousands of offspring. So you can see why it can become a little bit challenging working with particular organisms. So are all smooth seeds, again, masking this in our F1 generation, and then seeing that recessive trait reappear with this 3-to-1 ratio in our F2 or filial 2 generation. Masking the F1. Okay, so we know dominant alleles mask recessive ones. This is something we've learned about for a long time. And this is where we're getting those distinct segregation ratios. So different genotypes, it's important to be really careful with words here. Our genotypes refer to the collection of alleles that we have, what's going on with the DNA. So this could be homozygous dominant, so we have 2 dominant alleles. You could have heterozygous, 1 dominant and 1 recessive, or homozygous recessive, 2 copies of the recessive allele. This is the genotype. Okay, so when we talk about genotype, we have to worry about what the alleles look like. So we'll see some different ratios with the genotypes. When we talk about the phenotype, we're looking at the physical trait these genes are controlling. So in this case, smooth seeds versus wrinkled. So the phenotypic ratio here is what we're talking about, the 3-to-1. So we can revisit Mendel's experiment, and we can see our smooth versus wrinkled seeds. Okay, in the F1, you can already tell what the dominant genes will be. All smooth, all yellow, all tall, green, inflated, all purple. You can really see what's happening there. That's going to be our dominant allele. And then when we cross an F1 by another F1, we get the F2. And we can actually see the number of recessive coming back out again. And then the dominant to recessive ratio in that F2 will then be 3-to-1. So 3-to-1 ratio, this is about where we're sitting. Okay, and again, this is the phenotypic ratio that we're looking at. So we actually, you have to remember that Mendel couldn't get in there and actually sequence these genes. So he's looking at the phenotypic ratios here. So from these data, Mendel proposed his first law, the principle of segregation. So in this particular principle, recessive traits, which are masked in the F1 from a cross between 2 true breeding strains, meaning homozygous, reappear in a specific proportion in the F2, that 3-to-1 phenotypic ratio. What he really is getting at here is the idea that alleles separate during gametogenesis. So this is meiosis. This parallels homologous chromosome separation. So if you remember back to chapter 12, we talked about homologous chromosomes separating. This is where the alleles separate. So alleles for genes are kept on homologous chromosome pairs. When they separate, that is when we get allele separation and you can get dominant or recessive. These results traditionally are recorded in a Punnett square. We'll see some other ways to do this today. So thinking about how the Punnett squares are set up, I think it's important just to make sure we're able to do this really precisely. So we have parent 1, in this case our female, parent 2, our male. Parental phenotype, smooth seeds. Parent 2, wrinkled. Okay, and we're talking about true breeding strains here, which again implies homozygous, dominant in this case, recessive for our wrinkled seeds. Okay, And this is the diploid parent. This is what the parent has going on in all of their cells. And these are homologous chromosomes. So these two chromosomes here are homologous. These two here are homologous, and they happen to have the same allele here. That doesn't mean that they're necessarily completely identical all the way up the rest of the length of the chromosome. But we can go ahead and think about during meiosis, we're going to end up going from this diploid 2N state to a 1N state and separating these homologous chromosomes so that each gamete only gets one. Here are haploid gametes. So parent 1 could give us dominant S or dominant S. Our parent 2 can give us recessive S or recessive S. Okay, so now what we're looking at is how to set the Punnett square up from these gametes. So parent 2, this is just on the top. One gamete goes here, the other gamete goes here. For parent 1, one gamete goes here, the other gamete goes here. So it's really important to keep those gametes separate. And we'll be doing some examples of Punnett squares where we're actually looking at more than one trait at once. So it'll be important to keep those straight. And of course, the Punnett squares are just going to show us what happens, kind of a what if each of these pairings occur. So what if parent 1 gives us this particular homologous chromosome and parent 2 gives us this particular one? This will be the resulting genotype of the offspring and the resulting phenotype. You can see there's not much to say here because everything, we have true breeding parents, so everything will be heterozygous in the F1. Okay, and again, this is going to parallel homologous chromosome separation in meiosis, which we can just kind of get a visual on that, remembering homologous chromosomes, and they're attached to their sister chromatids here, so that kind of complicates things. Okay, so we're going to have our orange homologous chromosome, our blue homologous chromosome, and they will be lining up and then pulled apart during anaphase 1. Okay, so that's when the alleles separate. In this case, somebody could get a dominant allele, somebody could get a recessive allele. So again, we can see how these will go out. Okay, so we can have this case, we're now looking at making the F2. So this looks like it's the same Punnett square, but it isn't. This is going to be an F1 by an F1. All of those F1s on the previous slides were heterozygous. So heterozygous by heterozygous. Now we can have a chance of giving a dominant or recessive for either parent, and you can see that reflected in the setup, either dominant or recessive for both parents. Now we get to see where these ratios come out. So you can see in this case, we have one individual who is homozygous dominant. So this would be what you would call true breeding. It shows a dominant phenotype and will always give dominant offspring. We then have one individual who shows the recessive phenotype. This is also true breeding, or it's going to be homozygous for the recessive allele, two recessive alleles. And then we have a pairing of one dominant, one recessive. This is going to be heterozygous for both of these individuals. The phenotypic ratio we see here is smooth, 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 wrinkled, 3:1. The genotypic ratio is 1, referring to our homozygous dominant, to 2, these two heterozygotes, to 1. So that's our homozygous recessive. So as you get more comfortable with genetics, for now we kind of need to do Punnett squares just to double check what ratios we'll get. But as we become more comfortable, these ratios will become second nature and we'll be kind of internalizing them so that we're able to recognize deviations from them. So these results can be represented by a Punnett square, or we can do something called a branch diagram. So as we set up this branch diagram, it's going to look like it's more trouble than it's worth. for this particular cross because we could easily do this in a Punnett square. Where things like branch diagrams really come in handy are when you start to deal with multiple traits. So we're kind of working our way up to that type of analysis. And this type of diagram uses principles of probability to predict phenotype. So we're getting the same information as a Punnett square. Ultimately, maybe not in this first case because it's a very simple cross, but in more complicated cases, this can more easily give us those predicted phenotypes. So first, what we're going to do to set this up, and now in this particular case, we're going to be looking for the genotypic ratios. So first, we must list the probability of passing on each allele for parent 1. So for one parent, you have a 50-50 shot. I either give big S or little s. Okay, we write those down separately. So 50-50 shot, 1/2 big S, that goes here. 50-50 shot, small s, that goes here. We do the same thing for the other parent, but we have to do it twice to account for the scenario where we could get a big S from the first parent and then either a big S or a little s from the second parent, or we could get a little s from the first parent and either a big S or little s from the second parent. So this is where the branching actually occurs, okay? Similarly, this parent was heterozygous, so there's a 50-50 chance of passing on either the big S or the little s. That's where the 1/2 comes in. So again, doing this twice to account for each of the possible combinations. So the next part of this, we're going to multiply. So this uses principles of probability. The way I like to think about this is when you're looking for the probability of the outcome, you list the things that need to happen. And if things are like, let's say I want to know what's the probability of getting a big S true breeding parent, then I would expect that the situation would be one parent has to give a dominant allele and the other parent needs to give its dominant allele. When you hit that big and, that means multiply. So the probability that we'll get a true breeding dominant offspring or homozygous dominant offspring are going to be the probability of one parent donating the dominant allele multiplied by the probability of the other parent giving that dominant allele. So we can see how that works out. So 1/2 times 1/2 gives us 1/4 true breeding homozygous dominant. And again, in this particular example, we're specifically honing in on the genotypes. In a lot of the examples later on, we're going to be focusing only on the phenotype, so the ratios we expect will be a little bit different. If we want to know how many offspring, what proportion of offspring are going to be heterozygous, we have two ways to get that. Either the first parent gives its dominant allele and the second parent gives its recessive allele. 1/2 times 1/2 is 1/4. Or the first parent gives the recessive allele and the second one gives the dominant. 1/2 times 1/2 is 1/4. And you can add these together so you have half a chance, 50-50 shot of getting a heterozygote. Likewise, recessive, 1/2 times 1/2, 1/4. That's going to be basically that 1 to 2 to 1 ratio that we looked at for the genotypes. So you can see how that is going to play out. If you want to look at the phenotypes, you can see that this will be a 3/4 dominant phenotype and a 1/4 recessive phenotype. So in the problems, we'll be very specific about indicating whether we're looking for the genotype or phenotype. Again, this seemed like it's really complicated for this setup. This will make your life so much easier when we start dealing with 2 or 3 or more traits at one time. So Speaking of that, what if we have more than one trait? Okay, and it's very important here to be really respectful of the difference between the allele and the trait. So alleles are kind of within the same gene, the same trait. Alleles are variations on a trait. So if you think kind of hierarchically, a trait would be up here, and then you'd have multiple alleles that could result from it. So for example, the trait we've been working with is smooth versus wrinkled. That's controlled by one gene, and we have alleles, dominant or recessive, within that gene. The second trait, in this case, yellow versus green, has its own alleles that can either be dominant or recessive. So in this particular case, these genes are on completely different chromosomes, which is great for Mendel's work. We'll get into that a little bit later, okay? So in this case, we're going to have parent 1, which is going to be homozygous for both smooth, homozygous dominant for both smooth and yellow seeds. And it's going to be recessive for parent 2 for both the wrinkled phenotype and our green seeds. So these are going to be true breeding homozygous parents, one dominant, one recessive. This is kind of an abridged Punnett square because from doing previous ones, we know the only thing that parent 1 can give is a dominant S and a dominant Y. So we're able to just kind of condense that into a shortcut because otherwise you could just draw this 4 times. Then we know our second parent will be recessive for both traits. So again, we've kind of condensed this. So our F1 will be heterozygous for both traits. So this may start to get confusing to you, but remember that each gene kind of operates within its own Punnett square. Each trait operates within its own Punnett square. So if you had no problem realizing what would happen in the F1 and F2 generations for the smooth wrinkled trait, you could just do a Punnett square, figure it out. You could kind of do your own separate Punnett square for the yellow-green trait, and they're operating kind of within their own set of Punnett squares. So this kind of thing is referred to as the principle of independent assortment. And what that means is that getting a dominant seed color gene is not at all dependent on whether you got the smooth or wrinkled allele. So they kind of do their own things, again, operating within their own Punnett square. So in our previous example in meiosis, the relationship between homologous chromosomes is kind of a horizontal relationship. Like these two are going to be homologous chromosomes. These two down here, paired, are homologous chromosomes. You can see that they separate. Independent assortment is the relationship, or the relationship between traits is more of a vertical relationship. It's the relationship between this set and this set. So these are two different chromosomes, two different traits. And the idea is that, yeah, in this case, orange and orange went together, but it could be orange and blue going together, or blue and orange. So whether you get an orange for the first chromosome doesn't control what happens with the second. And that's the whole underlying idea with independent assortment. So it's really important to make sure that you're really straight on what an allele is. That's, again, the relationship between homologous chromosomes. That's where alleles are. And traits, which at this point presumably are on different chromosomes, the relationship is this independent assortment. So we can think about all kinds of things. And the more gene pairs or traits we have, the more phenotypic classes, and look how many genotypic classes. So if you just have 4 traits, you have a possible 81 genotypic classes with 16 phenotypes. That's going to be a heck of a Punnett square. Okay, so this is where the branch diagrams do start to come in handy. So let's just explore one of those Punnett squares so you can see the kind of heinousness it will actually give you. So this is that same cross we saw before. Okay, this is going to be our F1 generation. All of the individuals in the F1 are heterozygous for both traits. So now let's do the F2. So F1 by F1, heterozygote, heterozygote by this double heterozygote again. Okay, man, look at the combinations you could draw. All right, so we're going to take a second just to outline where these could come from. So in this case, our first gamete happens if dominant S segregates with dominant Y. The second gamete is what if this dominant S came along with a recessive Y? Or what if recessive S came along with dominant Y? Or what if recessive S came along with recessive Y? Okay, so these are the options that can happen. There are 4 for each parent. And so all 4 of those are lined up along our Punnett square. So then, oh my gosh, tired myself even looking at this. Then you have to say, okay, big S, big Y, dominant, dominant. Big S, big Y, little y. And at the end, you're like, why am I even doing this? Okay. And you can get particular ratios for this cross. You'll get 9/16 with the dominant phenotype for both, 3/16 for one dominant and one recessive trait, in this case, smooth green or wrinkled yellow. And then one individual out of 16 will have recessive traits at, or recessive alleles at both loci or both traits. So this is a lot of work and gives you a lot of opportunity to make mistakes. Okay, can you do it this way? Sure. But now imagine that you've got 3 traits instead of just 2. You can imagine the expansion that would happen there. Okay, so again, we can think about these different crosses. This just kind of gives you a summary of the different crosses you could do and different phenotypic classes. But you can see if we could shorten this, this would be a lot easier. So let's try this as a branch diagram. And it does take a little while to get the hang of this, but once you do, you can do this simply by multiplying without having to set this whole branch diagram up once you're really comfortable with the ratios. But for now, we're going to go through it step by step because that's how we learn. So branch diagrams make it easier because they allow us to deal with each trait separately. So here we have that same F1 by F1 cross that was in that big Punnett square. So the first thing that we do, in this case, unlike our previous example, we're interested in the phenotypes. So we can write down the proportion of expected dominant and recessive phenotypes in the offspring we would expect for trait 1 only. So let's deal with only the smooth wrinkled trait first. Let's ignore that the yellow-green thing exists. Okay, so I kind of think of this again as handling this within its own Punnett square. We have a heterozygote for this seed morphology trait, heterozygote. If you did a heterozygote by heterozygote for just this one trait, phenotypically, you'd end up with 3/4 smooth and 1/4 wrinkled, okay? So we've basically taken care of that Punnett square. Now let's do that for the second trait, the yellow-green trait. But again, we have to do this twice to account for independent assortment. So again, it's possible that a dominant phenotype assorts with a dominant or recessive phenotype, and that a recessive phenotype for trait 1 would assort with a dominant or recessive. So that's why we're writing this down twice, okay? It does not matter which trait you do first. The first one, you write it down. The second one, you always have to write it down to account for the possibilities. And yes, if you've got a third trait, you would have another trait or branch where twice here, or write it down here, 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 4 times. So each time you branch this off, you have to account for both possibilities for the remaining trait. So now we've got 3/4 smooth, okay? And then we've got a possibility of 3/4 yellow, 1/4 green, because this was heterozygous, okay? 3/4 yellow, 1/4 green. This was, again, this heterozygous thing, we've written it twice. Now, if I ask you any of these things, what are the probability of getting a wrinkled green plant? What is the probability of getting a smooth yellow? What is the probability of getting a wrinkled yellow? All we have to do is multiply. So 3/4 times 3/4. Okay, we can do all of that. 9/16. We can get 3/4 times 1/4. It's 3/16. 1/4 times 3/4. 3/16. 1/4 times 1/4. 1/16. Those are those same exact ratios we got from that big Punnett square. So again, practicing with these, you won't even have to write all this out. If you know what's dominant and recessive, you can just know if it was a heterozygous cross that it's going to be 3/4, 1/4, and then multiply by whatever you need to for the next one. Okay, so we'll get some practice with this in your assignment. Okay, so the important thing about these Mendelian ratios is not just that we know them. So while they're pretty cool and it's really neat that somebody tested all these plants to figure this out, the main reason that we need to know these ratios is to know if something deviates from them, because that could mean something weird biologically is going on. Okay, so in the next part of this slide set, we're going to talk about the chi-square test. Okay, so the chi-square test will tell us if something deviates from these Mendelian ratios. So generally speaking, the chi-square test is just a statistical test that will allow you to test for differences, whether if you have this null hypothesis, whether anything that deviates from that is due to chance, or whether we're going to see something meaningful causing deviations. Okay, so in order to do this, again, the essence of this is knowing what to expect from these ratios and comparing what we expected to what we actually see, and seeing if that difference is large enough to be something we should explore further. Okay, so we need a few values here. Okay, to do this, we're going to need what we expected, we're going to need what we observed, and then we'll need a couple of other details as we go. So this is the chi-square formula. We'll be working through this. It's one thing I'd like to just reiterate many times is that chi-square is the name of this test, and it's unfortunately named because this is not x squared like in just regular algebra. There's some algebra involved, but this is not solved for x. So we're not going to square root this at the end. So just get that out of your mind right now, no matter what. And I'll keep telling you that because it's a problem. Okay. So first, let's think about how we get each of these frequencies. So we know from this cross, we're working with a similar cross. Okay. This is going to be smooth yellow, smooth green, wrinkled yellow, wrinkled green. And I think it's important to note here what this cross actually is. So I don't always love the way that your book actually sets this up, because they don't always tell you what the crosses are right out in front, but it's always important to know what your cross was. In this particular case, what we're looking at is going to be a heterozygote, a double heterozygote, crossed to a homozygous recessive plant. So this is like a test cross that we're doing here. Okay, so this will be what we're looking at. I'll leave this up until we actually get to maybe a point where it's going to get in the way. But for now, let's leave that up here. This is the cross that happened, and these are going to be the results that we're looking at. Okay, so the observed, how do we get the observed? man, you just look at them and count. Okay, so these data, these numbers have to be given to you in some way, shape, or form. You can't just logic through the observed numbers. So somebody sat here and counted 154 smooth yellow seeds. Somebody counted all of these and got a total of 568 and wrote down the numbers. Okay, that's where the observed come from. You have to be given that data, or at least the opportunity to collect that data. So how do we get the expected frequencies? And this is where knowing the cross that we set up is so important. Okay, so this cross here, I'll go ahead, let's get that little marker back out. Okay, so this is the cross. How do we get the expected? It's from Mendelian ratios. Okay, so basically the idea is, for this particular cross, we're going to have possible gametes for this parent. Could be big S with big Y, big S with little y, little s with big Y, or little s, little y. These are the gametes for that Punnett square. Okay, I'm doing this with my mouse, so try not to judge me too hard for this. Okay, for this guy, luckily we can do an abridged Punnett square because the only thing you can give is a little s and a little y. That's the only possibilities. So little s, little y. Okay. All right, and I know this is going to get in the way, so we'll eventually erase it. But for now, you can have, and this is, I'll just put a dot under my big S's so that you know. Okay, so this is big S, little s, big Y, little y. This is going to be big S, little s, little y, little y, little s, big Y, little y. Okay, and then little s, little s, little y, little y. Okay, all right. So phenotypically, smooth yellow, smooth green, wrinkled yellow, wrinkled green, 1 to 1 to 1 to 1, or each one of these is 1/4, 1/4, 1/4, 1/4. And you can do the whole thing out, 16 squares, you'll get the same thing. Okay, so let me just erase this. And this is why it's so important to know the cross. And then I wish that your book would be a little bit more upfront with, hey, this is the cross, because this is how you get the expected values. So if you have a cross other than this, your expected values will be different. Okay, for now we know 1/4, 1/4, 1/4, 1/4, smooth yellow, smooth green, wrinkled yellow, wrinkled green. Okay, so let's get rid of all this mess before it gets in our way. This is a video, so obviously you can replay it if you weren't quite ready. I'm going to erase this. Okay, so our total was 568. We know that this will be 1/4, 1/4, 1/4, 1/4. Divide this by 4 and split it evenly. 142, 142, 142, 142. These are the expected values. So you have to be able to figure out Mendelian ratios to know what you expect. The observed is just observed. This is the thinking part, the expected. But once you have that, all you have to do is plug and play in this equation. Okay, this is observed, that's the green one. This is expected. Okay, so let's do the actual rest of this. So observed minus expected for each of these. Okay, and this is the sum, but we'll worry about it at the end. And your book or this table also refers to the observed minus the expected as D, which you'll see in this table in a moment. Observed minus expected, 12. Observed minus expected, minus 18. It's okay to get a negative. That's why we're going to square it. 144 to 142, 2, 146, 142 is 4. Literally just observed minus expected. Okay, and that gives us what we call D, the observed minus the expected. Do this for all 4 separately. Don't try to get all fancy and add anything yet. Now, we calculate D squared, which is simply D, these values you've just calculated, squared. 12 squared is 144. 18, even negative, 18 squared is 324. 2 squared is 4, and 4 squared is 16. That'll be the next set of values in our table. And then now we can take our observed minus expected squared and divide it by the expected. Okay, notice again, we're doing each of these individually. We're going to sum them at the end. So let's do that last part. We got D squared over the expected. So D squared over the expected. Got D squared over the expected. Okay, doing this for each of these. These are the values you should come up with. Now we can add them. Okay, so we got all these. Now we can add them. This is what we sum them as. Okay, so this is going to be our sum. All right. Chi-square, sum this all up, 343. Okay. So we've got this value. What is this? Okay. So please, I am begging you. First of all, notice I never took a square root. So I'm going to remind you about that again. Chi-square is a value you get. You can get a very large value for this. You can get a really large value. Don't be tempted by it. Don't try to make yourself more comfortable by doing a square root to make it smaller. Leave it as it is. Okay, so this is the value. This value tells you nothing on its own. Don't tell me about some significance of this value. This is not directly a discussion that we're going to need to have. In order to actually convert this into any kind of probability, we're going to need a little more information. Okay, so specifically what we're doing here, notice this. You know, sometimes math makes sense. Remember that the overall idea of what we're doing here is trying to figure out if our observed number is very different from our expected number in a way that should alert us to something going on. So that's what we're doing. We're looking for the differences between observed and expected. Okay, so all this tells you is kind of an indirect measure of how different they are. This does not tell you whether that level of difference means anything yet. Okay, so this is just at this point, an arbitrary number. So in order to figure that out, we're going to need one more piece of information, and this is called the degrees of freedom, which if you've taken statistics, this is something that you've heard about before. For the chi-square test, the degrees of freedom is the number of phenotypic classes, 1, 2, 3, 4, minus 1, 1, 2, 3. So this is 3. The degrees of freedom is 3. Our chi-square value is 3.43. Armed with that information, we can now actually learn something. So our degrees of freedom is 3, which means that we ignore everything else on this chart except what's in this row. You can see that there is a threshold. This is at the 0.05 level. If you're more comfortable with a 0.01 level, your threshold will move to the right. Again, remember that as these increase, indirectly it's telling you that your observed and expected are getting further apart. There's a bigger difference. So you got to set a threshold to know where you're going to make the cutoff or how different is too different. Okay, so our value was here, or somewhere around here, 3.43. Notice it's not going to exactly fall in this chart. You got to kind of guess where it's between. Okay, Now there's this threshold, which here is set at 0.05. Okay. What this means is that we're going to fail to reject our null hypothesis. Okay. So in this case, the null hypothesis would mean that our offspring segregated in the expected Mendelian ratios, and anything that deviates from that was simply due to chance. So if you see anything different from an exact 3-to-1 ratio or whatever we're looking for, it was due to chance. So if we fail to reject it, that means that the differences we saw were not large enough to be significantly different. They're probably due to chance. If we reject the null hypothesis, so we get a larger chi-square and we're sitting somewhere over here, depending on where we're most comfortable, that cutoff, what that means is that deviations that we saw from the Mendelian ratios are large enough that, statistically speaking, something other than chance is happening. So something biological underlying them, and in this case, we reject the null hypothesis. So this means something interesting is going on. What is that interesting thing? Man, I don't know. It could be several things. It could be that genes are linked, which we'll be talking about later on. It could be there is some lethality associated with one of the alleles. It could mean that there's transposable elements at work. So there could be a lot of things causing this. We don't know what it is yet. What we know is that it's different enough that biologically we should explore it. Okay, so things I don't want to hear. I don't want to hear about a 3.43 being statistically significant. Don't give me that. It's got to be at this threshold. This is our like p-values up here. We want to know if we meet that cutoff or not. In this case, we did not. So everything we saw that was different was just due to chance. Okay, so we'll be using the chi-square test a few times in later chapters as we start talking about linked genes. Okay, so mathematical, man. All right, so the last sections that we're going to be talking about are going to be pedigrees. So we've seen these before in the paper we read on the first day of class, which we'll be revisiting. But pedigrees are something that we often use when we can't make controlled crosses. So with those plants, who cares if we rip out the stamens of one plant and like intentionally put pollen from another plant on it? They're just plants. We cross them all day long. All right. So with humans and animals, you know, you can't do things like that, which is fortunate for us as individuals. For genetics, it makes things complicated, but we can kind of see how things plinko out. So this particular pedigree, this is going to be, again, generations. So this would be like grandparents. Okay. So males are depicted as squares. Affected individuals for whatever trait you're talking about. In this case, we're talking about albinism, which is going to be a trait that we see when we have a mutation in a melanin producing pathway. So our males are depicted as squares. Affected individuals or those who have albinism are depicted, in this case, as purple. Usually it's a shaded or not shaded, but each pedigree will have its own key. Okay. Females are depicted as circles, and then expected offspring can be depicted as diamonds. You've got twins. These would be identical twins, note the little like offshoot, and then fraternal twins. Okay, so we've got different kind of things going on here. Really, what we need to know is kind of what's going on with this. Okay, so pedigrees can be used in various ways. Sometimes when we don't know if the trait is recessive or not. Okay, so with something that's recessive and autosomal, we can see that we're going to be affecting males and females equally. So I don't have an example here, but sex-linked traits, if you remember from chapter 12, these are going to affect males more often. So here we can see males and females affected. That tells us it's not linked to the X chromosome most likely. And we should see fewer individuals, more, this is not exact, but closer to that 1/4 ratio if we're looking at a recessive trait. Okay, being able to mask that. So note all of these offspring are masked. Here we have a situation with, I would call this, you know, this is like identical twins, which they're not the same person in their soul, but DNA-wise, they have the same allele for albinism, so you don't necessarily have to count those as like. This would be not that the albinism happened twice. This would be kind of like a once and split. Okay, so again, thinking about this type of thing, you can figure out that something is autosomal recessive by looking at the pedigree. Okay, with something that's going to be dominant and autosomal, you're going to see something that maybe not exactly, but it's going to, again, if it's autosomal, will affect females and males the same. And you're going to see that we're going to get a lot closer to that 3/4 ratio. In this case, it looks like about half of the offspring are affected, but a lot more than that 1/4. Okay, so again, it's harder to tell in people because we didn't have thousands of offspring like the plants did. Okay, so we don't know how that would have plinkered out eventually, but we'll get closer to that 3/4 ratio. So in your assignment, we're going to be asking to calculate the probability that the offspring of two parents will show a particular trait. So to do this, this is going to be a very general example, but it should get you through sort of the initial part of this assignment. Okay, so being able to figure out the genotype of our parents to begin with. Okay, so being able to figure out the genotype of the parent is kind of step one, and we can do this based on the pedigree. And then we have to understand the probability of that parent passing on this trait. Okay, so we'll use this autosomal recessive trait for albinism to do this work. So first, being able to figure out the genotype of the parent based on the pedigree. So let's use individuals 2-4 and 2-5. Okay, they have a dominant phenotype, but you can figure out that they both have recessive alleles, so they're heterozygous, both of them, because otherwise you wouldn't be able to have homozygous recessive offspring. Okay, so these two guys here, you can see they're homozygous recessive offspring. Additionally, individual 2-4 has a homozygous recessive parent. So this particular parent can only pass on a recessive allele. So this person has to be heterozygous or what we call a carrier. So a carrier is a person who does not display the trait themselves, but has the recessive allele so that they can pass it on. So this person is a carrier, okay, because they have a recessive allele. Again, because we see recessive offspring from 4 and 5, we know that individual 2-5 also has to have a masked recessive allele hiding in there somewhere. Okay, so this is how we figure out whether they're carriers. And we're going to also learn how to do probabilities for those if we don't have enough information to go from. Okay, so let's also talk about whether they'll pass it on. Okay, so the probability that an offspring will have a particular phenotype is going to be based on both the probability that the parent has particular alleles, so are they carriers or not, and whether if they are a carrier or do have that allele, whether they actually give it to the offspring to begin with. Okay, so let's start with the probability that the parent has particular alleles. So we're going to use individual 3-5. The question here would be like, if individual 3-5 had an offspring, what would be the probability that they would have albinism or they would have the recessive allele? So notice We can't be entirely sure about their genotype, their status, because they could be homozygous dominant. We don't know if they even have that allele. So we're going to have to calculate probabilities in the absence of any information about their offspring. So to answer this, we need to use Punnett square genetics, and we're going to get probabilities. So in order to think about individual 3-5, the question we're asking is, what is the probability that this is a carrier? We have to think about the Punnett square that made them. Okay, so this is 2-4 and 2-5. We know 100% these have to be carriers based off of their offspring and parents. So this would be their Punnett square. This is 2-4 and 2-5, heterozygous, big A, little a, big A, little a for albinism. Okay, so these genotypes are the resulting offspring that could be 3-5. However, here's a pro tip. What's the phenotype of 3-5? We already know it doesn't have albinism. It does not display the recessive phenotype. So we can go ahead and nix that out and just ignore it. So the probability that it's a carrier is going to be, here's a carrier, not a carrier. It's going to be 2 out of 3 or 2/3. So this person has, there's basically a 2/3 chance that this individual is a carrier. Okay. So now that's the first part. The first part of this is, do I have the allele? And then the second part of this is, do I pass it on? So this part's easier. In a diploid system, if you're a carrier, I have big A, I have little a, 50-50 shot. Either I give you big A or I give you little a. So that's 1/2. So if we want to know if what the chances are of any offspring of 3-5 going to be to give a recessive offspring, 3-5 would have to be a carrier and pass down that recessive allele. That's that big and, which means multiply. So 2/3, which is the probability that it's a carrier, times 1/2 equals 2/6 or 1/3. So this is this parent's probability of having and passing down a particular allele. Of course, you have 2 parents, so you need to do this for both parents. We didn't use 2 examples, but the general problem or general sort of equation is the probability of parent 1 having the allele times the probability of them passing it on, which is for diploid organisms pretty much 1/2. Okay, unless you're going to be homozygous. The probability that parent 2 has the allele times the probability of passing it on, that's going to be the general equation. So you guys will be able to practice this in your assignment. To conclude this chapter, we can see the genotype. It's going to provide a blueprint for the phenotype of an organism. So we never want to lose sight of that connection, no matter how technical we get, between genotype and phenotype. Mendel's first law tells us that alleles segregate independently. Okay, so this is going to be pulling apart during meiosis 1. This is homologous chromosomes. And that different traits, 2 different traits, are independent of each other, of course, unless they're linked, which we're going to be talking about later. And using these ratios, we can predict the outcome of the offspring produced, which is going to, again, give us some higher level genetic skills, because if we can predict the outcome, we can do a lot of things related to deviations from that outcome, and we can calculate some probabilities.